Okay, we're going to take a look at blood typing today, and then we're going to look at the genetics of blood typing. And I thought a nice way to do this would be to look at the blood types in terms of just food coloring. Now, of course, blood is not these different colors, but I thought it'd be a good representation of the different blood types. If we represent this one as blood type A, we'll represent the blue one as blood type B, B and B, and we're going to represent O as the clear water. Now, O is a good example of clear water because of the fact it has no proteins on it. In other words, I can give blood to anybody. For example, I could give blood to B, and B stays B, right? It doesn't change. I could give O blood to A blood, and again, it does not change. That's why we call O blood the universal donor. In a minute, I'll show you what that looks like. Now, my question to you is, could we give A to O?
No.
No, right? If I put red in with the clear, it's going to become red. Now, the body is going to defend against that in terms of it's going to produce antibodies, and my body doesn't recognize those if I have O blood. The same is true for B. Now, what's the fourth blood type is AB. And I got this to represent AB blood. Now, AB blood is called the universal receiver, right? What I did is I put some red and blue food coloring in this glass. And the reason it's called universal receiver is because it can receive from A, does not change the blood type, can receive from B, also doesn't change the blood type, and of course it can receive from O, and it still doesn't change the blood type. Now, so we have four blood types. We have A, B, O, and AB. Now, of course, the next question would be, what about the plus and the minus? Mr. Trevelyan and I have O negative blood, or Mr. Trevelyan and I have A negative blood. I would like to represent those as ice cubes. Now, the Rh factor is different in the sense that it's a different gene on a different chromosome. And if we represent this ice cube as the Rh factor, which is what the positive and negative is, it stands for rhesus monkey, by the way. If I wanted to make it positive, I could just put an ice cube in. Now I got A positive blood. And if I wanted O positive, I could put an ice cube in there. Now, the problem with the Rh factor is once there's an ice cube in there, it does change the blood. And I cannot give somebody who's Rh positive with the ice cubes, I couldn't give that, for example, some of that blood to B negative, because now B negative would have the positive antigen in there. It would produce antibodies on it and would kill that individual. On the other hand, if I had no ice cube in there, if I was Rh negative, O blood, universal donor, I could give to A positive because it doesn't recognize anything different. It doesn't change the blood, right? So if the ice cubes are representing the Rh positive individuals, who do you think the universal receiver would be? Who could receive from anybody? Now, we said that purple, right, represented AB. We said that A could give to AB. We said that B could give to AB. And we said that O could give to AB. What about in terms of the Rh factor? Well, if I was to give this guy with the ice cubes to AB negative, it would change its blood type, right? So I can't do that. The universal receiver is going to be AB positive. I could give A negative to this person. That would be okay. I could give B negative. I could give O positive or negative, right? Anything can go in here because he has all the antigens. He has the protein, antigens are proteins. He has the protein for A, has the proteins for B, and he has the proteins for the Rh factor. Who would be the universal donor? Who could give to anybody?
O negative.
O negative, right? Nope, no ice cubes in there, right? I could give to this guy, doesn't change his blood. Give it to this guy, doesn't change his blood. Give it to this guy, doesn't change his blood. So if we talk about negative as the universal donor, we could talk about AB positive as the universal receiver. Let's take a look at this. I think we mentioned earlier that the blood types, and we'll get into this eventually, but the genetics behind the blood type is we said that A and B are dominant over O, but dominant traits don't always give us the most common trait. For example, in this case, O in most races, O is the most common, even though O is negative to both A and B. A is the second most common, and so on and so forth. The rarest blood is way up here with AB negative, right? The least amount of people have that. If you're trying to get blood from somebody, boy, AB negative is going to be tough. Now, this is what I'm trying to show you in terms of these antigens. Antigens are proteins, and they're on the blood cells. So the red circle in the center is representing the blood cells. And if you look closely, you'll see the difference between those. Now, they don't have little A's on the blood cells, but A blood has little A triangles, and B blood has those little B hearts. And if you're AB, of course, you'll have the B hearts and the A triangles both. If you are O blood, notice that the O blood has nothing on it. And the same is true on the bottom. If I'm O positive, now I get nothing on there except that Rh factor. If I'm A positive, you'll notice I got the A triangles plus the Rh, little green squares that are going to make them positive. If I'm B positive, notice it's got the Rh factor. That's what makes it a plus, and the little B hearts. And if I'm AB, notice it's got the A's, the B's, and the Rh factors as well. Now, the reason blood is tough to give to other people are because of these things called antibodies. These are the things that attack foreign substances in a cell. So if I have blood type A, I produce antibodies against B, and I can't give blood from an A person, from a B person to an A person. They would produce antibodies, their blood would clot, and it would actually kill them. The opposite is true for blood type B. It has antibodies against A. If I try to give A blood to a B individual, the antibodies will kill them. AB, remember, has no antibodies. We call it the universal receiver. It can receive blood from AB, and O because it recognizes both the antigens for A and B. And again, there's the O on the bottom. I like to consider it a naked blood cell because, again, it has neither antibodies against A or B. So we call it the universal donor. Now in lab today, you're going to use these little dishes. I'll show you in 1 second. And if they clot up inside these little dishes, we say that they have the antigen against that particular blood cell. So if I'm trying to test somebody's blood, if it gathers up like so and clots, this would be blood type A. Inside that tube was a little bit of the antibodies. And when I put the blood in there, it clots up, which means he has blood type A. Now it also clotted here at the Rh factor, which means he's A positive. The second individual, well, didn't clot in the A antibodies, but it did clot in the B. So he's B, and he's B negative because the Rh antibodies also did not clot. How about the third individual? Well, didn't clot here, but the Rh factor did clot. He has O positive blood. And down here, you'll notice that it clotted in the A and the B, but not in the Rh factor. He has AB negative. All right, your turn to try. Now, these are the blood cell. These are the little dishes you're going to get. Again, I'm going to show you those in a minute. And if you're remote, you're going to watch somebody else do it. If you're in lab, you'll be doing this yourself. You're going to put a little drop of blood in each little container. Careful where you're putting them in, and be careful that you don't mix one with the other. If you contaminate that, then the antibodies are going to be in both containers, and that's going to be a problem. It's not going to give you good results. But anyway, let's take a look at these four. In the first one, it's clotted in the B and it clotted in the Rh factor. What type of blood would that be? Would that be A, AB, B, or O? And would it have the plus or the negative if it clotted in the Rh factor? The second column there, we see that it clotted in the A and the Rh. What type of blood would that give us? In the third column, there is neither clotting in the A or the B, nor the Rh. What type of blood would that be? And finally, in the last one, you'll notice that it clotted only in the antibody A. What type of blood would that be? Let's take a look. Did you get them right? The first blood type is B positive because it clotted in the B and it clotted in the Rh factor. The second blood type was blood type A positive, clotted with the antibody A and with the Rh factor. The third column, none of them clotted. It didn't clot with the antibody A, it didn't clot with the antibody B, and it didn't clot with the anti-Rh antibodies, so we call that O negative. And in the fourth column, you'll notice it clotted in the A only, not the Rh factor, so we call it A negative blood. All right, now, if we go back to our original slide, let's take a look